Okay, so I got so carried away with angiosperms the other day, and class ran a little bit long. You guys are actually, about a third of a chapter behind the other class, but that's fine. I don't care because I like talking about this stuff. So again, don't worry about the schedule and all that. I'll keep us on schedule. We'll get all the material done. I mean, come on now. I'm a zoologist. We're not going to gather this class without me spending my time talking about the critters, talking about the animals. Okay. Well, we left off with the idea of angiosperm evolution, okay? And this is another one of those things that bothered Darwin. Remember Darwin's understanding of the fossil record, even though he had the mechanism of natural selection down right, the fossil record was confusing to him because, of course, it's incomplete. It's still confusing to us now. But one of the things that really bothered him was when it wasn't complete. In other words, when it seemed like nothing was going on, and all of a sudden, in a narrow piece of rock, which Darwin and geologists knew that was time, there was all these critters that were coming, all these fossils, right? And then it would settle down a little bit, and then it would do it again. Remember, that was the Cambrian explosion. Well, another thing happened in angiosperm evolution. Not quite the explosion that Darwin thought of. Remember, he had an incomplete fossil record, so he sort of thought that they went from no angiosperms to angiosperms in this really short period of time. And what we find out is that, angiosperms were always already showing up long before Darwin knew they were showing up. The fossil record was incomplete. They'd only traced angiosperms back to, I'm not sure how many millions of years ago. But we know now that angiosperms are actually a lot older, and it started evolving long before Darwin knew that they were. As a matter of fact, if you look at the one in the lower right, the one on the left is Archaefructus. Isn't that a cool name? Archaea fructus. Archaea is primitive or first, original, like the domain Archaea, the extremophiles. And it was an interesting looking plant. It looked like something you might see today. In the upper right, I found an artist's, it was an aquatic plant, right? You can see the fish swimming around there. I found an artist's picture of what it might have looked like. So it kind of looks like it had these little pineapple things at the top, but they weren't pineapples really, because we're not quite to that point yet. But on this thing, we had the carpels, right, the female part. We had the stamen. So we got the cones that we've been familiar with. And we even think that these little things in the fossil record here, I don't know how well you can see with the picture there. There's another part of one right there, were floats. So this thing, that's why we think it was aquatic. That's why botanists think it was aquatic. But even further back in time, there was another one that we found called Montsechia vidali, this one right here. This one we believe was even older than Archaefructus. The Archaefructus was the one that we thought was the first, or one of the very first. But we think that this one might have been first. And what you're looking at in the lower picture, it actually had seeds with coverings, fruit-like things. So, I mean, maybe we weren't exactly an angiosperm, but, unbeknownst to Darwin, this little thing down here, right, that the oldest known angiosperms in the fossil record, these guys here, there's a lot of doubt as to whether those are the first. Probably not the first. So it really wasn't an explosion. It was, in the Cambrian explosion, both of these things were explosions, okay? But they weren't quite as quick as Darwin thought they were, number one. And he also, remember, did not understand or did not have a grasp of cladogenesis then. Remember, cladogenesis is the branching. It's the punctuated equilibrium. And he didn't have that wasn't a part of the evolutionary theory back then. That wasn't until the 70s, remember? Okay, so let's look at some groups of different types of plants. This is this plant here in the upper right, which is sort of an interesting looking plant. It looks like it's almost candied. You know what I mean? Like candied fruit or something that you would eat. It doesn't look like it's got a sort of a candying to the outside of it. Well, this is Ambarella, and Ambarella still is alive. It lives, of course, in one of those weird places in the world, northeast of Australia. So these little islands. I think it's the New Caledonian Islands. I think that's where Guadalcanal is, where they fought one of the battles in World War II. New Caledonian Islands. I believe your book tells you where it was. New Caledonia, something like that. Anyway, so it's a secluded place off by itself. And remember, we talked about what happens when you're off by yourself, that sometimes you can kind of get away. That's kind of the whole thing about Australia, the land that time forgot. The whole continent of Australia, the landmass, separated from the rest of them a long time ago. So those weird critters that you find in Australia, the reason why they're so weird is because they lived in this small little, relatively speaking, continent all by themselves for a long time. So they kind of took evolution, and when they were there, the original ones that were there before it split away were very stem organisms. So they had a lot of places to go. They were, in terms of the evolutionary sense. So Ambarella is probably the most recent common ancestor of all the living angiosperms. So that would be, again, what we would call a living fossil, right? Not something that's lived, but not like a coelacanth, right? Coelacanth we thought was dead, but 65 million years ago, but it isn't. But something like the whisk fern, remember? The whisk fern looks a lot like Cooksonia. Doesn't mean that they're necessarily related. Although Ambarella here, this one that you're looking at here, Ambarella trichopoda, it is the closest living relative to the original angiosperms, but it's not necessarily in the direct lineage. All these branches off, right? Oh, what else we got here? Monocots, dicots. We talked about this in lab the other day. We used to use this as a taxonomic separation. There was, I don't know what it was, a class or whatever it was, but there was a monocot taxonomic group, and there was at that time, dicot. Now it's called a eudicot taxonomic group. And we thought that meant something about relationship. But as you can see in my little wording there, these groups, monocot and dicot, are paraphyletic. They're paraphyletic. So the differences that we see between them have nothing to do with the fact that they're related to one another. So there's a lot of convergent evolution going on. Okay, the dicots or the monocots are named that because when they're first a seedling, and I showed you this in the reproductive thing the other day, how many what we call embryonic leaves, how many first leaves do they have? If they only have one, then it's a monocot, that leaf being the cotyledon. They're really kind of cool words, C-O-T-Y, cotyledon. If you got two leaves, two of those embryonic leaves, then you're a dicotyledon. Right, so you get two of those. Makes sense, doesn't it? Well, got it backwards from what I had before. Whatever. The eudicots. That's the most speciose. Speciose means the most numbers of species. 170,000. These are trees, shrubs, the things, legumes, beans, lettuce, oak. Things you're the flowering things that you're very familiar with. With 170,000 species, most of the things that you look at are probably dicots. Now, monocots have not far, they have what, about half, a little under half of the number of species as the eudicots. But agriculturally speaking, and for your yard actually, these are some of the more important plants. These are the grains, wheat and rye and the things that we eat, the bananas, palms, right? Even a palm tree, the sable palm is not a tree. Okay, it's a big, really thick piece of grass. That's all it is. It's a thick, and then of course, like I said, your lawn grass is a monocot. Typically, monocots don't make really big flowers, and if you've ever seen your flowers on your palms, they're really kind of small little things, aren't they? They're not very big at all. But if you grab one of those pollen, where all that pollen is found, you shake it, it makes this yellow smoke everywhere. See, monocots use wind for pollination. So they're going to make a lot of really light pollen that float with the wind. That's why they don't have very big flowers. There's that detective part that I've always been talking about. If you were examining a flower, someplace you'd never been before, and you noticed it was rather small, like sometimes they're so small they're not microscopic, but they ain't very big. They're a few millimeters in some cases. So sometimes you're fooled going, well, I've never seen this bush flower. Does that mean it's not a flowering plant? It looks like a flowering plant. Yeah, it makes really small little flowers sometimes. But if you looked at a flower like that, you could probably say, well, it's probably not using insects for pollination. See, the ones that use the insects for the pollination, those are the flashy ones. Those are the trees. Those are the eudicots. Okay? So you can tell a lot by just looking at those sorts of things. But sometimes you get fooled because monocot and dicot, like I said, were originally thought as taxonomic groups. But the characteristics that we're going to go through here, that I'm going to show you, we went the lab, you had a little handout, or not a handout, what was that, a little case? These characteristics that unite them aren't there because they're necessarily related to one another, okay? So we already talked about the cotyledon. If you're monocot, you have one, let me write one, two if you're a dicot. If you're a monocot, the veins, the xylem, the phloem, the tracheids that are on your leaves are parallel. And if you're a dicot, they're net-like. They branch out. If you're a monocot, your vascular tissue, all right, your xylem and your phloem, if we took a cross-section through a stem and we looked at it, those vascular tissues are scattered throughout the pith. Somebody asked me the other day, what is the pith? The pith is, if I took the inside of a tree, ones with rings, or the inside of a big, if I cut one of those palm trees down, which would look like a tree because it isn't, that stuff on the inside is the pith. Some plants are very pithy, which means they're very stiff. Some plants aren't so pithy. Pithy always also means to be kind of catty. Like, oh, aren't you pithy? Or does it mean to be clever? I never know. Nobody uses that word. It's like the word tardy. I was thinking about the other day. Does anybody use the word tardy after they get out of grade school? Do adults ever say, hey, man, I'm sorry I was tardy? Okay. If you're a dicot, though, your vascular bundles, your vascular tissues are arranged in a ring. Okay. So dicots are trees. So those are the rings they're talking about. If you look at a tree, everybody knows the rings in a tree, nothing spectacular, but those are those vascular bundles. The reason why some of the rings are darker and some are lighter is because the tree is always adding to the outside. It's always growing wider. But in the summertime, it grows wider faster, so it adds rings faster, and there's more space between them. And in the wintertime, it grows more slowly, and there's less space between them. So if you looked under a microscope, those dark rings, or what we call rings, are actually a bunch of little tiny rings close together. And then the summer ones are further apart, and then they're close together again. Everybody knew this, right? You learned this back when you were in grade school, didn't you? Please tell me you did. Okay, the roots are different. If you're a monocot, it's basically you don't have this thing in the middle, this thing called the tap root, or my dad always called it the sucker root. If you don't get the sucker root out, you won't kill it, whatever we have to pull out of the ground. But the tap root, that's you find that in dicots. Monocot, if you look at the pollen, so this is going to be a little hard out running in the field. Monocots have one opening in the pollen. The pollen grains and dicots have three. The bottom one is probably, well, to me anyway, this one, the leaves, the way the leaves look, right? And then counting the floral organs. And what we're talking about is the sepals, right? Those green little leaves that are underneath. The petals, the stamen, the male part, and if it's got more than one carpel, remember not all flowers have more than one carpel, the female part, but if it's got more than one of those, how many does it have? And if it has these floral organs in threes, 3 petals, 6 petals, something like that, then it's a monocot. And if the multiples are in 4 or 5, then it's a dicot. Okay, so monocot 3, dicot 4 or 5. Now, be cautious with that when you're trying to identify flowers. Very often, if you're counting petals, petals fall off. And very often, when you're counting stamen have fallen off. So counting sepals is not a bad way to do it. Sepals are the little leaves underneath, and they're a little more durable. They don't fall off as easily. So I often don't count the sepals. But I would. That's why a botanist, if he's trying to do that, will count a lot of flowers on the same area that look alike, just to make sure he's not got one with missing a petal. Hey, you're missing a petal. All right. Now, let's go back into this idea of evolution of angiosperms or how they're related. There's about 100 species of what we call basal angiosperms. Your author or the authors of this book don't even give you the taxonomic classification for angiosperms. They don't give you the phylum and classes in order. It's really just kind of too complex. So they're going to simplify it for you. There's no taxonomic classification called a basal. So we're talking about the base. We're talking about those that were some of the that are related to some of the first angiosperms, right? The things like the water lilies, Ambarella, which we just talked about, the living fossil kind of thing, the star anise. We have a water lily here in Florida. So we have a basal angiosperm. It's this one up here in the upper right, the American white water lily. I've seen them at Venetian Gardens quite a bit. I've seen them lots of lights. Okay, so those are the basal angiosperms. Now, one of the bigger groups of angiosperms is the magnolids, named after the, in taxonomy, they have, like, if you're talking about the group magnolids, whatever that might be, then you would say that all members of this group share a characteristic that are sort of, that link them together as magnolids. And you're going to name the group after typically who you think was the first magnolid. So if the first one in this tree group wasn't magnolid, but it was Ambarella, then you wouldn't call them magnolids, you call them Ambarellas. Okay, so magnolids means magnolias, things related to magnolias like pepper. That's what's in the upper pepper grove, a pepper vine. Okay, that's a magnolid. The orange in the upper right, Persea americana. That's the orange. And then of course, the magnolia, one of my favorite trees. Love magnolias. Look at the name of magnolias. I love how they name things sometimes. See that? Where do you think that comes from? Why do you think they called it that? Grandiflora, absolutely. They smell great. I have one in my backyard. I thought the hurricane last time was going to blow it over, but it didn't. It survived. But okay, the monocots. But 25% of the species, right? It's the eudicots that's the big group. But the monocots, I said, are very important agricultural plants. Some examples of monocots again are palms, cabbage palm that looks like a tree, but it's just a big old piece of grass. Orchids. We have native orchids here in Florida all the way, and by the way, all Florida orchids are protected. FS is a Florida statute. It's illegal if you're out walking in the woods and you find orchids, and you might. I've seen them before. I saw some in Lake Louisa Park one time, but the next time we went back, I took my field trip there, it was gone, so somebody had snagged it. Your St. Augustine, that's a monocot. So grasses, those kinds of things are monocots. The eudicots, that's the big group, right? The legumes. So not all of the agricultural plants are monocots. Legumes are very important. The swamp rose, which has such a pretty awful, swamp rose for a pretty flower, Rosa palustris. She's a swamp rose. He's a swamp rose. Just kind of an awful name. Oh, one of my favorite, Florida Alecia, Chapmania floridana, the yellow thing up above. Florida Alecia, which you can find on campus, I've seen on campus tons of times, is really, really unique. And what's unique about it is it's Florida Alecia is an example of organisms that we say are endemic. Okay, so you see in the little map there, in the green is where you find Florida Alecia. So what do you think it means when we say something is endemic, like endemic to Florida? Yeah. Melanie, not necessarily from it, because we could say that was where it started, but then it spread out. But yeah, it's only found there. That means I'll bet you've seen this flower. I know you have. If I see some on campus this semester, I'll take you out and show you. But you just probably saw it on the side of the road. It's just some flower growing on the side of the road. And you cross the border into Georgia, and they're not there. You notice in the dark green toward the edges, they start to kind of filter out. But isn't that neat? Nobody in the world. So now here's what that says about protecting species in peril. Somewhere down the road, maybe somebody's going to launch something where they want Florida Alicia. And people are going to say, well, is the Florida Alicia population in danger? And they're going to say, well, no, the Florida Alicia population is stable. The numbers aren't changing. And you can say, well, why do you want to protect it? Well, sometimes we protect things because they're rare. Not because of the fact that where they're found, there's not a lot of them, but maybe more context of they're rare in terms of where else they're found. You see what I mean? Florida Alicia is only found in Florida. That might be something to think about when we talk about protecting species. And if for no other reason that species diversity is important for the ecosystem health, there is this philosophy. There was a guy named Popper. I read an article from him a long time ago, some sort of a paper, where he said that organisms don't have to prove their worth to us as humans, that they have worth simply because they exist. So things out there don't have to be beneficial or provide something for us, food or otherwise. Even mosquitoes, I know, pain is the butt they are. Even mosquitoes have the right to exist, okay? So sometimes we'll protect something only because there's just, not very many of them in the world. I mean, there's a lot in Florida, but that's it. We eat a lot of seed plants, obviously. A lot of our grains like wheat and maize, potatoes, sweet potatoes, my favorite. Sweet potatoes with honey butter and cinnamon. Oh, man. And then my mother used to cut them up and put marshmallows on top at Thanksgiving. Oh, I eat so many of those, I make myself sick to my stomach. That's what we eat mostly. This little part down here, what this is telling you is kind of interesting about what we eat on Earth. Basically what this says, to produce 1 kilogram of beef, a little over 2 pounds, to produce 1 kilogram of beef, in other words, to be able to raise a cow and to get a kilogram of beef from it, for every kilogram of that cow, you've got to feed it 5 to 7 kilograms of grain. So what you get out of the cow is, 1/5 to 1/7 of what you would have gotten out of the grain. And we're talking about energy content here. So the point of fact is yes, we do need protein, folks. So big thinkers, big brained organisms, humans do have a necessity for protein. But sometimes perhaps a little bit of too much is the problem here. And this is showing you that in many cases, all over the world where we have livestock for folks who are basically starving, who are having trouble feeding everybody in the area there, have livestock when actually what they should need to be doing is growing crops, because you get much more out of growing crops than you do out of growing beef. Of course, you have to keep in mind the protein, obviously, but you get that from someplace else. And a lot of the medicines, especially the ones, well, not especially the ones, but a lot of the medicines that are plant-derived are beneficial because they don't have quite the side effects as a lot of the other pharmaceuticals do. I always get a kick out of that. You guys are probably too young to watch the news every night, but my wife and I do. And you can tell who the demographic is. Oh, my God, every commercial is a pharmaceutical. Everyone. My son is so funny. He was sitting there watching, and he's like, he didn't understand the Cialis commercial. And the Cialis has 2 uses: erectile dysfunction and can't control your bladder. Is there a name for it? If men have difficulty holding their bladder when they get older, right? Cialis is good for that. So my son was like, well, that's a really nasty commercial. I said, what do you mean? He goes, well, they're talking about sex and then it's like, or stop to find a bathroom. And he's like, what, you gonna go have sex in a bathroom? Is that what they're saying to do in there? I'm like, no, it's 2 parts. Cialis helps you if you have erectile dysfunction. It also helps you if you what's that term called for being really drunk is one of the ways I know that happens, but nonetheless. So anyway, when you get older, maybe you'll experience that. See all these things like Taxol, ovarian cancer drug, curare. Curare is what the South American Indians would put on the tips of their blow darts. And look it's a muscle relaxant. So if you OD someone, I guess that is what it's called, right? Overactive bladder. My dad always hated my mom and I on road trips because my dad was like a camel. He like, all right, here we are in New Jersey. We need to drive to Texas. Anybody got to pee? You're like, yeah, maybe. He'd drive the whole way. Tiffany, what if you OD'd something on a muscle relaxant? What would help? Or what would help? What would it do? What if you got a big dose of muscle relaxant? I mean, to make you a little bit, might help your muscles relax, but what's the problem? I mean, if you were, yeah, that's right. You certainly wouldn't want to. Yep. Yeah, exactly. Yeah, you'd be sedated, right? So your muscles are involuntary and voluntary. The ones you want to move are voluntary. The ones that help you breathe are involuntary. But a muscle relaxer would relax them all. Don't want to relax the ones that help you breathe. No, don't do those. Okay, so that's put on the tip of their blowtarts, and when they would hit an animal with it, some of their prey, it would cause the animal then to die from that, obviously. Habitats to plant diversity or threats to plant diversity. Habitat destruction, number one reason why things go extinct, folks. Don't ever forget that. Number one reason why things go extinct is because you remove their habitat. So we have to think about that when we start to engage in species survival plans. Is there enough habitat to support the organism? We believe right now that within the next few centuries, if we continue at the present rate, that we're going to have a caused about the extinction of half the species on Earth. And this is not just plants, but this is all species on Earth. When it comes to plants, one of the places we're doing that the biggest is, of course, in the tropical rainforest. I'm sure you guys have all heard about that, where we clear an area about the size of West Virginia every year. Look at the before and after of Brazil down below, 75 to, 2012. On the left is the dark green is tropical rainforest. On the right, the dark green is still tropical rainforest. All that other stuff, agriculture. Here, city, agriculture. Most of the tropical rainforest is gone. But another 200 years, if we continue along those lines, we won't have tropical rainforest anymore. What does that do? Well, that's a lot of trees absorbing CO2, and we got a problem with CO2. I remember once I was having a discussion with somebody, and they said, well, what's the problem with CO2 increasing in the atmosphere? We wanted to increase plant growth. And I'm like, no, you can dump CO2 in the atmosphere a lot faster than plants can grow and suck it up. And if you're cutting them down to the size of West Virginia every year, 55,000 square kilometers, you're really exacerbating the situation, aren't you? You're removing the trees or the things that take out the carbon dioxide, and yet you're still putting a lot more in. And therein lies what we have with global climate change. Okay, I'm not going to go over this last slide. It's sort of a study slide for you. I would take a look at it. It's a good way to do a little kind of a compare and contrast study between the different groups on some of the major characteristics that we talked about, okay? I'm going to move on to the next chapter now. Where's my mouse? There we go. We're now going to talk about probably one of the strangest organisms on the planet, these guys. I say strange because plants live different than animals. Animals live different than plants, and there are some big differences between them. But honestly, if you were to ask the person on the street, mushroom, oak tree, or human, which of those is least like the others? What do you think they would say? So the Sesame Street thing. So mushroom, oak tree, human. Which one of those is the standout? Which one is the out group? Good guess. Good guesses. Yeah, I would too. Nobody's gotten it right yet, though. Yeah, it's of those, it's the plants. Mushrooms or fungi in humans are more closely related to one another, just by a little bit, just by a smidge, but more closely related to each other. In other words, where we branched off that big family tree was a little bit after plants branched off from the branch that gave rise to animals and fungi. And there's a couple of evidences that kind of point to the similarity between, or at least to the connection, and I'll get to those. One of the reasons I like a fungus is because they can kind of really do whatever they want. They can be whatever kind of color they want, all kinds of beautiful patterns. They don't photosynthesize, and they can grow like this Lactarius indigo. Lactarius is because if you were to break this open, it has a latex, like a white liquid in it. Okay, it's kind of sticky too. The stuff that you see on the tree there, that's called turkey tail, right? That's another one that's kind of interesting. Now, a lot of turkey tails look, there's a lot of variation in them the way they look. Turkey tail, because it looks like, like when a turkey's got his tail open in the coloration. Those are Florida. You might have seen some of those. All right, so we dispelled one misconception about the relationship between human and fungi and plants, right? I would have guessed the same thing, honestly, before biology. I would have went with you guys just what you were saying too. I would have thought, honestly, that it's the fungus and the plant that's probably more closely related because they kind of look like they're doing the same thing, even though one's not photosynthesized. So here's another one for you. I went and gave a talk in a middle school many years ago, of course, the kids. Who would win between a shark and a killer whale? I'm like, oh, I don't know. They always want to know that kind of stuff. But one of the questions I got was, the kids said, what's the biggest organism in the world? So I said, organism or animal? And he goes, oh, animal, I meant. I'm like, all right, so it's the blue whale, right? Blue whale's the biggest one. And then I went back and I said to the kids, I said, but that's not the biggest organism in the world. So we talked a little bit about organism and animal and all that sort of stuff. So I'm going to ask you guys, what do you think is the biggest single organism in the world? And again, think along the lines of the biggest animal being blue whale. That's a good guess, Harrison. Those sequoias are really, really tall. Redwoods out, they get what? 100, 200 feet tall, something like that? Not the whale, right? Because, blue whale will get 100 feet long, something like that. Yep, they can get a lot bigger. Yeah, well, that's not right either. It's neither a tree, it's neither the sequoia, it's not a blue whale, but it's a fungus. Somewhere out in Oregon, oh, do I have it still pulled up? Let me see if I can send you the link. Oh, yes, I do. Somewhere out in Oregon, there is the world's largest single organism. I'm going to put it in the link in the chat box. Look on that. And what we mean by that is in this, so where is it out in Oregon? I read it earlier this morning. It is Blue Mountains in Oregon. It's the world's largest organism. It's a honey fungus. Now, the funny thing about honey fungus or honey mushrooms, or maybe not the funny thing, but the neat thing is they're here in Florida. Here, this link, the second one, those are ringless honey mushrooms, which you might have seen and probably have seen. Honey mushrooms are very common. Very common. As a matter of fact, most times if a student shows me, Mr. Clark, what mushroom is this? Remember I told you, I'm not really big on fungi. If it has this honey color, I usually go, try honey mushroom. That's usually my response. But these common mushrooms and, Armillaria, that's the genus for these honey mushrooms. There's more than one species. But there is a species of this particular type, this particular species that covers 9 million square meters of forest. 9 million square meters. It's what, 3.5 miles across? What did it say in that article again? 3.4, 2.4, 2.5 miles across. Over 1,900 years old, weighing hundreds of tons. Hundreds of tons. That's the world's largest single organism. Now, the reason why you might be going, wait a minute, I can't imagine walking around the woods in a toadstool, a mushroom that could be 2.5 miles across. Well, that might be because of your misperception about mushrooms. Most of a mushroom's growth is underground. Okay, when you see these things, these toadstools, what's happening is the mushroom these are called fruiting bodies, reproductive structures. The mushroom is reproducing. The mushroom says, oh, conditions are right. And sometimes when we talk about organisms reproducing, because conditions are right, the conditions could be either good, changing, or they might be bad, right? It depends on the species. It just Kind of depends on the organism itself as well as the specific conditions. All right, so what is happening with this big mushroom is this growth that's occurring underground. Now, that's what's 2.5 miles across. That's what covers 9 million. I think I called my class this morning 9 million square miles. No, I think that's more than the Earth. 9 million square meters. I hope they figured that out by now. 9 million square meters. It's the underground part. You notice, like, when you wake up in the morning, sometimes perhaps you look in your yard and go, hey, where'd all the mushrooms come from? They were always there. They were underground. It rained overnight, great conditions. Boop, all these things pop up, these toadstools. There you go. Now you know. We're related to mushrooms more than we are to plants. And the biggest critter in the world is a mushroom. The honey mushroom in, where was it? Blue Mountain, Oregon. 2.4 miles. Holy cow. Okay, it's a fairly speciose group. You know, the insects, they're going to win all the time. Look where we are. We're over here, by the way. We're a tiny, look like even the tiniest sliver of the organisms out there. Fungi make up a fairly decent part of the pie. About 1.5 million or so species of fungi, so they're pretty species rich. They come in many different types. They can be single-celled or multi-celled. Again, not quite the same multicellular we see in animals. And when we get to animals, I think we'll eventually get to clear that up, what I've been meaning by that all along, as I keep saying that. I'm talking about animals in the form of tissues. Reproduction can be of both types, sexual or asexual. Now, here's something very unique about them. When we digest our food, we digest our food what we call intracellularly. And that basically means inside of us, all right? We have enzymes in our stomach, we have enzymes in our intestines, and that helps us digest the food. You all know all that kind of stuff. But what fungi do, and this kind of goes back to that mat that I'm talking about that grows underground, all that stuff that grows underground, they digest stuff using exozymes. Exozymes are enzymes, but they're enzymes that are used on the outside. So they digest their food extracellularly. So that little mat that we're going to look at that crawls through underground, it's secreting all these enzymes, right? And it's causing all this organic material in the dirt to be broken down. And then it can be broken down simpler and simpler and simpler, and eventually you can use it for food or you can recycle it. That's why mushrooms, fungi are good recyclers. They digest them. Remember we said that's how plants got out on the land, right? It's going to be tough living on land to begin with. But plants moved out there because they had a fungus, remember the mycorrhizae? A fungus that grew around their roots. And that fungus helped them liquefy the organic material and made the soil richer. So the plant had a better chance of surviving. Very important on Earth that we have these decomposers. They're not the only one, but very important because they do that. They recycle that material. Well, there it is. There's the years. 460 to about 670 million years ago. It's hard to tell. Somewhere in there, we had a common ancestor, somewhere around that time. Bracketing, interestingly enough, the Cambrian explosion, when a lot of these critters came into being. All right, let's stick with this nutrition thing for just a few more minutes. This is a bracket fungus, and a bracket fungus is not a species. It's just a turkey tail that I showed you as a bracket fungus. It grows like on a shelf, right? Bracket fungus. Okay, so we talked about what an exozyme is, and that underground mat that we're going to look at here in the next couple of slides in more detail. That underground mat is called a mycelium, and it has a very high surface area. It actually has a very high surface area to volume ratio. And if you remember back from Bio 1, when you figured out why cells are so small, you said that cells are small because they're more efficient at moving things across the membrane if they have a high surface area to volume ratio. And that happens when you're small. Now, mycelium, it would be great to have that high surface area to volume ratio because that's what they do. Remember, they're absorbing materials from the outside world. They're absorbing that stuff that those exozymes are liquefying or digesting out in the dirt. They can break down things like cellulose. You and I can't do that. Cellulose is, plant cell wall material. Cows can, but they have those symbiotic bacteria, right? You and I can't. We can eat cellulose. It's roughage. If you ever like look at what is it called, Metamucil, the ingredient Metamucil is cellulose. It's just meant to clean your pipes. And lignin. These guys can also digest lignin. I think we talked about that's lignin. Lignin is one of the most durable organic materials on Earth, and it's very wealthy, rich in carbons. So it would be, and it's a major component of trees. And what allows trees to really be hard to stand up. So it'd be a shame if that was lost when the tree died. Well, very few organisms can break down lignin. It's like really hard glue. Mushrooms can do that, or fungi can do that. Yeah, and some of them, they're carnivorous. Carnivorous mushrooms invading, walking around, attacking things. Wouldn't that be interesting? That'd be a good. I'm sure there's been a really horrible sci-fi movie about that before. I remember as a kid living in Germany with the German woman that lived downstairs from us, and she would take me out the woods gathering mushrooms because she would cook with them, right? And it was always very adamant. She always was very adamant. Never put the mushrooms you pick, me, into my bag. Only put them in your bag. Well, and the reason why is because I didn't know which ones were poisonous, and she did. So she never used any of the ones that were in my bag, obviously. I never knew. I just knew it was very rude to put your mushroom into another person's bag. Okay, let's kind of dig into this, what they're using to get nutrients out of the environment. So this underground mat is made up of these things called hyphae, and hyphae are filaments, okay? They can either be made of many cells, or they can be made of one cell, and that might be a little confusing. At least it is to me. How can an organism be a cell but be considered multicellular? Well, that's because there's another weird thing about fungus. In the fungi world, it's not about the cell. Okay, in the fungi world, it's not about the cell. It's not about cellular reproduction. It's not about the cell doing this. It's not about the cell doing that. It's all about the nucleus. See, so what happens? Look down below. See those two types of hyphae, the septate and the synocytic? Synocytic, that's how you say that word. The septate has septum, spaces between them. Now, there's pores, and things can go through those pores. For example, in the picture you see there, you can see things could pass through. So the little purple dots that you see, those are nuclei. They could pass through. But look at the hyphae on the right. There's no divisions. All those purple things are nuclei. So what you have in a situation among some fungus with the synocytic hyphae is you have what we call a multikaryotic of structure, many nuclei. Remember, the nucleus is really the important part of the cell because of the fact that it contains the DNA, and life needs the DNA. Nobody ever said that you had to put that DNA in a nucleus into a cell. In this particular case, the fungi decided to do something among some of them a little bit different. They made one large structure with many nuclei in it. Now, I'll talk in just a minute, maybe some of the advantages and why they would do that. But one of the reasons why this whole either synocytic hyphae or the septate hyphae with the pores, one of the things that makes that really good is that's how they can grow really fast. Remember I said they've all popped up? So all that mycelial, all these little hyphae are growing underground. It rains that night and they go, ooh, and they start pumping cytoplasm into their hyphae, and they can do that really quick. Even if they have the pores, they can do it really quick, and they plump it up, and the toadstools come up out of the ground. All right, now here we got to get the bigger picture. Each one of these little things is the hyphae, okay, and this is the mycelium, the mat. All of that. This is all mycelial mat. This is, look at 60 microns. Look how thin this stuff is. Right? Lots of absorption can go there. And again, the thing that pops out of the ground, what is it? It's hyphae too. It's just hyphae that have been swollen by the cytoplasm flowing into them. If you've ever taken a mushroom, a raw mushroom, or maybe the next time one's on your salad or something like that, grab it by it's not really called the stem. Grab it by that part right there and pull it. It'll tear apart like in fabric. It doesn't. I can't. What am I trying to say? You can tell that it's layers of like these stringy things. It looks like it tears apart like masses of string would tear apart. Okay, those are all little hyphae, and they're all with this massive amount of surface area because they're secreting all these exozymes. You remember the thing about these guys is it's all about digesting food on the outside, so maximizing that surface area to volume ratio. So the hyphae, do that. They obviously act a little bit like roots. They provide some stability. It's not hard to pull a mushroom out of the ground, but look at all that. This is them at 10 microns. And then I got a couple other pictures. It looks like somebody just splattered paint on the wall. In the big background, that's a mat of hyphae. Look, I got two cool microscope shots. Here's a cynocytic hyphae. And then there's septate on the right, the purple one. Okay, now a little bit of detective work. We found out that the cell walls found in these hyphae are made of chitin. You might have remembered from Bio 1 that chitin was it's a carbohydrate, but it's a specialized carbohydrate. It's what makes the exoskeletons of many creatures, many arthropods like insects and crustaceans, like this rhinoceros beetle right here. That exoskeleton is made of chitin. So there's kind of an interesting little light bulb right there, right? If the exoskeleton of arthropods is chitin, the cell walls of fungi, they're not cellulose like in plants, they're chitin, and then among a bunch of other evidence, like, you kind of start thinking maybe there's a relationship there. Hey, does anybody know if that's a male or a female rhinoceros beetle? How do you know, Dustin? Yep. So knowing that that's a male when it has the horn, what kind of behavior would you predict? What kind of reproductive behavior would you predict in rhinoceros beetles? Or if you remember the terminology, what kind of sexual selection would you expect to occur in rhinoceros beetles? Well, I mean, there is the one type, the one you're talking about, the one where the women do the selecting. That's called intersexual. In the case of a rhinoceros beetle, they engage in what we call intrasexual. Remember, intra means within the group, and inter means outside of the group. That's one of the things when I first came to Florida, I got so not confused, but I was like, wait a minute here. There's an Interstate 4. Does it go interstate? It ought to be called intrastate, right? It only goes Daytona to Tampa. Okay, so that big horn right there is going to tell you that it's probably the males doing battle, right? Those horns are used to wrestle the other male around. So it would be what we would call intrasexual selection. Say that 3 times real fast. See a little bit of that detective work again. Those are cool looking beetles too. Oh, here's the carnivorous one. Nematodes, which are little worm-like things. Well, they are worms. They're not worms like in the earthworms that you think about. But they're little microscopic worms, and sometimes what they do is the hyphae of a fungus can actually trap these things, and very quickly too. So these are the hyphae, and they can close around in less than a second. So boom, it traps this thing, and then of course what happens after that is they just keep wrapping hyphae around it. So it's kind of like what a spider does, when it takes the silk, wraps it around the nest or wraps it around the prey, and then they start digesting the nematode. I wonder in nematode world if they tell their kids stories about monster hyphae that eat up other nematodes. It'd be kind of hard to think, I'm afraid of fungus. Daddy, check the closet, make sure there's not fungus in there before the little nematode goes to sleep. Fungus can also wreak havoc on plants too. What they do is they form these things called haustorium, and haustorium really is anything that kind of looks like this. Winds back and forth. And what you're doing again is you're increasing surface area. Just like you learned in Bio 1 why the inside of the mitochondria is winding back and forth. More surface area, You got more of those pumps that you learned about. So these Hostoria, what they're doing is increasing the surface area, and this is parasitic because it actually, in many cases, is going to puncture into the cell and start digesting the cell. So plants and animals are not immune to fungus attacking them, being parasitic, or in this case, this is more predation. But not all of the fungus are bad. The mycorrhizae, there's a neat picture of mycorrhizae. These are the roots of the plant and all that hair in between. Those are fungal mycelium. Those are hyphae. They increase the surface area, digest a lot of nutrients. So there's really good evidence of the importance of them. That's why 90% of all vascular plants, you know, the ferns and everything after that, have these mycorrhizae. Look up here in this picture. This plant on the left was sprayed with a fungicide, something that would kill fungus. The problem when you spray things like that, you know, it's like my wife had a fungus on one of her plants. I said, be careful where you spray. You don't spray it on the ground, because you spray it on the ground and it gets and it kills the mycorrhizae, you're going to stunt the plant's growth. So the fungicide stunted the plant's growth there. This one over here was grown, you know, without being sprayed. So with the mycorrhizae. On LSCC campus, LSSC, holy cow. On the Lake Sumter campus there, and I haven't seen them this year yet, but if I do see them, I'll get you guys and show. There's these things called Indian pipe. Indian pipe is a flowering plant. That's actually a flower. Small little plants, fully white. No photosynthesis. They can't photosynthesize. But they're an example of this really unique thing that goes on in life called a tripartite relationship. And what we mean by that, a tripartite relationship, in this particular example, the Indian pipe flowering plant, the roots of that flowering plant actually parasitize the mycorrhizae that are growing on the roots. So very often it's an oak plant, on the roots of oaks. So it's a three-way thing. You got the Indian pipe parasitizing the mycorrhizae. Now, it's a relationship. Parasitism would be a plus and a minus, whereas the mycorrhizae on the roots are engaged in kind of a plus and a plus, right? The mycorrhizae on the roots are helping the plant, but the Indian pipe is parasitizing the mycorrhizae. Life finds a way, man. What time is it? Oh, we got plenty of time. Good deal. Isn't that cool? Can you imagine trying to figure out how this thing lived? Look, that's a flower. Ain't nothing green. And you can't photosynthesize unless you're green, unless you're red algae, and it's definitely not a red algae. Remember, this is what I was saying when plants moved out on the land. They had to move out on the land with these mycorrhizae. So that's why we look today, it's no surprise that 90% of all vascular plants have these on their roots. Notice not we didn't say non-vascular. So those would have been the first ones to move out, but without the mycorrhizae or a preponderance of mycorrhizae, they wouldn't have been the ones that could continue much further. They could eke out a living in the wet land that maybe the moisture helped the bacterial growth, would help with composition or decomposition. It's a little different when you're living further from there. So maybe these vascular plants needed that. Okay, two types of mycorrhizae. You have the mycorrhizae, like you see in the picture here, that actually surrounds the root. Okay, you'll see it under a microscope next week. But this is the vascular tissue, and that coating on the outside, that blue-green, that's the ecto, because it grows on the outside, mycorrhizae. It surrounds the roots, and it penetrates between the cells. So that's one type of ectomycorrhizae, or one type of mycorrhizae. And the other kind is called arpuscular. And the arpuscular ones are kind of like that Hostorium that I showed you. They form what's called an arpuscule. And an arpuscule is, again, just a lot of surface area. And they move themselves in through the plant and actually can penetrate through the cell wall, but they don't go into the cell membrane. If a fungus penetrates the cell membrane, oftentimes it's a parasitic fungus. Remember, these aren't parasitic; these are mutualistic. They're trying to help each other out. You can see the arpusculars in this picture. Look at that right there. And they even send hyphae between cells. That's what that is right there, as I scribble on it. All right. Okay, now let's talk about reproduction in these guys. And this is where it gets a little bit bizarre. Let's start with the zygote over here on the right-hand side. The zygote is very short-lived, and we'll get to why that is in just a second, but it's the only really diploid stage. And in a fungus, like I said, it's really not about the cell, it's about the nucleus. So you don't have, you notice the diploid, the part in the red, fungi spend very little of their life as a diploid, like we are, or as like vascular plants do, you know, like ferns and trees and all those stuff we learned about. But now what I want to do, starting from the zygote, is back up for just a second. Whether we're talking about a fungus or any other critter, the act of fertilization involves two sub-steps, okay? They're highlighted here in the slide. The first one is plasmogamy, and the second one is karyogamy. Now, what those words mean is in plasmogamy, that's when the two cells, whether it's sperm or egg or anything else we're talking about, when the two cells fuse together, that's plasmogamy. Now, in, say, for example, mammals, humans, and animals in general, usually very quickly does karyogamy occur. So the cells fuse, right, the sperm and the egg, and then the nuclei from each of those, they're haploid, remember, they fuse, and we're off and running with junior. That usually happens right afterwards. However, in fungus, the difference between plasmogamy and karyogamy can be in time. It can take many hours, days, or many centuries. Why in the heck would this thing want to stay in this purple part, a heterokaryotic? If you're septate, for example, and you have a septum that divides it up into cells, then we call you dikaryotic. But if you're synocytic, the open ones, you're multikaryotic. Whatever it is, you have many different nuclei, heterokaryotic. Why would you want to stay that way? Well, because there's more than one way to get something accomplished. And remember, your genetic information is going to help you in evolution if there's variability. So being heterokaryotic or being dikaryotic, in the case of the ones that are septate, kind of like being diploid. And remember, we said back in evolution that being diploid means that you can hide future, perhaps, beneficial information, right? The recessive alleles that aren't showing up today for some reason. Remember the heterozygote advantage? Remember the folks with sickle cell anemia? So when you're heterokaryotic stage, you have basically the advantages of diploidy again, but even more so because you don't. I meant to look this up between the break and I forgot. I don't believe in the heterokaryotic stage the dominant and the recessive thing plays out. So in the heterokaryotic stage, you have a whole bunch of nuclei out there with a whole bunch of genetic information. Oftentimes, like I said, these organisms are going to go into this plasmogamy and karyogamy for a long time. And a lot of time what they're doing is they're doing that when the environment is deteriorating. You see, the environment's getting bad, or maybe sometimes the environment's getting good. They'll go into this heterokaryotic stage because in some species we'll look at, being heterokaryotic means you're tougher. You even look tougher in some of the species that we'll take a look. But genetically, you have all of this information because when you're doing the dominant and recessive thing, you only have half. You know, unless you're homozygous, or dominant, but you understand what I'm saying. All right, now let's crank the wheel forward. So we get the zygote, we undergo meiosis. What's the purpose of meiosis? What is meiosis meant to do? In any organism, you, me, fungi, not so fun guy. I'm going to go take away your Bio 1 grades. Where did you have my Bio 1? Where's Isaac? Well, he was on Bio 1. Yeah, variability, right? So we get variability out of that. And we get spores. And we call these, to differentiate them from the other kind that you see, we call them sexual spores. Remember, sexual reproduction involves one very basic thing. Two cells coming together. So plasmogamy and karyogamy are sexual reproduction by definition, mixing the genetics. The spores germinate. And let's detour off to the asexual for just a second. So the spores germinate, little hyphae start growing out. Then it forms a mycelial mat. And then if we're talking about like toadstools, then those spore-producing structures, oh, it rained last night. Look, they pop up above the ground, and now they start producing spores, but they don't do it through meiosis. They do it through mitosis. So what do you get out of mitosis now? Yeah. So you go variability with when you go through meiosis. Yeah, there you go. And you get the quantity because it rained last night and all these toadstools are going to pop up over the ground and go, wee, wee, make spores and just make lots of them. And there's a lot of them going to be clones because you're undergone mitosis. But back when you did meiosis, you already got your variability there. Just do it. Go. Try to find a place. Go on, I'll write later. If you find a place, you germinate. Okay, and you form another hyphae and a mycelial mat. Now, we don't have sexes in fungus, so we don't talk about males and females. What we talk about are strains, or sometimes even something a little less descriptive, like mating types. Sometimes they'll be called a plus or a minus mating type. You'll see what I mean when I get to some of the other critters that we're going to look at here. But whatever we call the mating types are attracted to one another by pheromones. Pheromones are kind of external hormones is the best way for me to explain them. They're chemicals that are released by an organism to signify some communication to the environment. A female that's coming into heat, female humans don't have a heat cycle. They're what's called estrus. They're in estrus all the time, but other organisms aren't that way. And when the females come into heat, that's when they're sexually receptive. The other time of the year is basically leave them alone. They release a pheromone in many cases out to the world to tell, hey boys, I'm ready to reproduce if you're the one for me. Okay. There's some thought about whether humans have pheromones. Yeah, I don't know about that. I'm not sure we can smell well enough to really. Maybe that's what people say. Oh, it's just something, some chemistry between us. Maybe that's what they mean. But pheromones not only attract them together, but they also help keep the ones that are related. They also prevent inbreeding. So chemical signals. I'm over here, dear. All right. So that's reproduction. Let's look a little closer at some species who engage in asexual reproduction. Many species are sexual and asexual. We do have what we call an artificial group. Sometimes it's even given a taxonomic name. In fungi, sometimes the word phylum is not used. Instead of kingdom phylum, it might be kingdom division. I don't know if Deuteromycetes is a division, but Deuteromycetes is really a fake taxonomic group. Deuteromycetes means imperfect fungi. Doesn't mean there's anything wrong with the fungus, but we classify fungus based on how they reproduce. So whatever structures they make when they're reproducing, that's how we classify them taxonomic. That's how we've decided that they're related to each other, is how they reproduce. Now, if we've never witnessed sexual reproduction in a particular species of fungus, for example, maybe it hasn't done it in a long time, right? Or maybe it just doesn't do it, then we put it in this group Deuteromycetes. And then if we ever in the future do figure out how it sexually reproduces, and we find out what kind of structures it makes, and you'll see what I mean when we get into the different groups here a little bit later on, then we stick it in that group. So it's like the junk drawer at home. Throw all that stuff in there until it, until you find some place for it. But now molds and yeast aren't necessarily in the Deuteromycetes. Some species are, molds anyway. But these are examples of fungi that are real good at asexual reproduction. So that's penicillium that's growing on the outside of that orange. Penicillium is so cool. You'll see it under the scope. It looks like that that Greek woman with the snakes, Medusa, coming out of her head. And each of those little things coming out is a line of spores. That's what those little circles are. And every time this is disturbed, like if you were to, like the wind would blow, oh my God, all these spores come pouring out. So those are molds. Yeast is an example of an organism of fungus that's really known for its asexual reproduction, for its budding, reproduction by fission. Hey, bud, here's the parent, there's the bud. Hey, this is the scientific name for this yeast. Saccharomyces cerevisiae. What do you think this yeast is known best for doing? Yeast do things? Based on its name, I think it's kind of neat. Sometimes they name things. Dustin, you had to have been to Mexico or into the Caribbean somewhere where they speak Spanish. What is una más cerveza? There you go. Cerveza, Saccharomyces cerevisiae, is the yeast that they use to brew beer. Look at all these facts you learned in your biology class. Isn't that something? Okay, ancestry. Where do these guys come from? Well, fungus, like a lot of critters, don't fossilize very well. The oldest the oldest fungi are somewhere around 460 million years ago from the fossil record, but that molecular clock that we talked about back in evolution, the thing that works like a metronome, the musician, tick, 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 it keeps time. The molecular clock tells us that animals and fungi diverged, that's a pretty wide range, somewhere about a billion to 1.5 billion years ago, plus or minus 500 million years. We do know that the ancestor was aquatic, it was unicellular, and it was flagellated. Okay, and that also is another reason why we put these together with the animals. So take a look at this chart down here. So here's this aquatic, unicellular, flagellated ancestor. Okay. Now, what we got going on here, here's the animals, and over here would be somewhere breaking off would be the plants. Now, it's not drawn on here, but they say the animals and their protozoan relatives. Remember, it was a protozoan that gave rise to animals and to plants. So you notice that animals and fungi are more closely related. Or wait, I'm sorry, the plants wouldn't come off of there. They would come off of down here. There we go. And then the fungi split off, and we'll talk about the different fungi when we get into them. But this group over here, including the animals, we're all lumped together under a group of organisms known as the opisthokonts. And the opisthokonts are those that produce cells with rear-facing flagellum, with posterior flagellum, like these sperm cells. And indeed, humans do produce, when they produce flagellated cells, we put the flagella on the back, like Euglena puts the flagella on the front. But that's what links us together with the fungus. So all fungi are opisthokonts. Now, the nuclearids that are right there, those are protozoans. So fungi are more closely related to protozoans. That group is then more closely related to animals. Some of them are actually more closely related to other protozoans. Remember? Protozoan group is a mess. It's all paraphyletic and mixed everywhere in there. It was the stem group that gave rise to all the other ones, though, remember? It was the protozoans that gave rise to the plants and the animals and the fungus and everything out there. So that's why mixed in with the fungi, related to the animals, is another protozoan, the nuclearids. Okay, so what we're going to do now is we'll start going through the different groups. We've got the chytrids, very small group. The zygomycetes, another relatively small group. A very small group, but a very important group. Very important group. And then the more familiar groups, what we call the sac fungi. Right, this is a sac fungus. It's not called a sac fungus for the reasons that it looks like in the picture. And then a club fungi. And it's not called a club fungi because it looks like a club when it's a toadstool, because not all club fungi look that way. Okay, let's go chytrids. About 1,000 species, the phylum or division sometimes, Chytridiomycota, found everywhere, even down around those hydrothermal vents. Even down in that place where we talked about the biogenesis of life. This is a pretty ubiquitous group also in what they do. They can be decomposers, they can be parasites. That's what this chytrid is doing here on the right. Odygonium is a green algae. Odygonium is the green algae, and that's a filament, and that chytrid is penetrated into that green algae, and it's parasitizing it. Dog eat dog, even down there at the cellular level, isn't it? The chytrids kind of went off their own way very early on. We know they're opisthokonts for sure because when we look at their spores, this is a microscope picture of their spores. They have a flagellum. They have a rear-facing flagellum. That's very unique for this group. They're called zoospores. The reason why is because zoo refers to animals, and one of the major characteristics of animals is the ability to move. Now, the chytrids, this is the hyphae, are the only ones with those posterior-facing flagellum. That's what I mean when I say it's unique to this group. That doesn't mean the other ones didn't have it. It means they may have lost it, or their ancestor had it and they lost it before they diverged off. Remember, that's what makes you all the opisthokonts. This is the black bread mold. Okay, or the stuff that gets on your strawberries. It's the stuff that, like I said, it grows on the bread. So with this one, I want to start out. Let's do the asexual first. The sporangium, these little lollipop things that face up. These are cool. We'll look at them under the microscope. Look at these over here. These are another species, but the reason why they look like an army, don't they? They can some species can actually move their sporangium and face them in a way where, oh, it's a little wetter over there. Oh, the wind's blowing, this way, or whatever the case may be. Very spooky, isn't it? But the sporangium, regardless of where they can move or however they're structured, that's where the spores are made. And that's asexual. That's going to be typical in these kind of molds, is asexual, producing a lot of spores. If the environment begins to deteriorate, then sexual reproduction. Here's where I got the mating types. Mating type plus and a minus. And what they're going to do is they're going to fuse. And they're going to form what's called a gametangia. A gametangia is what happens is the fusion of those two mating types, those two hyphae, right before the cells fuse, right before plasmogamy. Now, once they are in plasmogamy, what they do is they form this thing called a zygosporangium. That's what it looks like under the microscope. That's the part in the middle. And it even kind of looks like it's tough, doesn't it? It looks all gnarly and hard, and it is. It's metabolically inactive, so it's like it's like seed, right? Seeds of plants. It's resistant to freezing and drying. It's even got a tough outside, so it's resistant to crushing, within limits. So this zygosporangium, during this heterokaryotic stage, look, heterokaryotic is N plus N. It's not 2N, right? Notice in the red and the blue dots, those are all nuclei, but you have a bunch of nuclei all in the same area together. So it's heterokaryotic. You got all that nuclear information you can use during this environmentally deteriorating time. Ooh, we better save our pennies. We don't know what's going to happen. Now, as things begin to look up, like it looks all right, looks like the environment's getting a little more conducive, then karyogamy is going to occur. Days, weeks, centuries later. So then karyogamy occurs, the nuclei fuse, you form a very short-lived zygote because you immediately go into meiosis. Because what you're doing is cranking out variability. All right, so you're going to make that zygosporangium, and then when you think maybe there's a chance that some of your spores will survive, you sense the environment getting better, the environment no longer is deteriorating, then you're going to run it through meiosis, get that variability, send it out into the environment. Life of a fungus. Any questions on that? I got one more slide I want to go over today, folks. Any questions on this? About 1,000 species. Like I said, synocytic hyphae, molds, parasites, commensals. Again, they're classified in this group because of their sexual structure. So the structure they make during sexual reproduction is called a zygosporangium, so a zygomycete. So if one of those deuteromycetes was shown to have a zygosporangium, then that's where we'd stick it. Okay, the glomeromycetes. This is the one with about 160 species. Very small. At one time, they were lumped with the ones we just went over. But again, that's just because of similarity of appearance, and we know that's not always the case. The reason why I say these are small but a very important group, remember 90% of all vascular plants have mycorrhizae, right? You got to have them to grow for the most part. And 80% of those mycorrhizae are of this type, are of this arpuscular mycorrhizae. See the arpuscular? See the arpuscle right there? This is the cell wall. All right, but that's an arpuscle. And of course, this is the mycorrhizae again that increases the surface area and the absorption. Well, most mycorrhizae out there, of that 90%, are of this group, the Glomeromycota. And nearly all of them form this arpuscular, not the ectomycorrhizae that grows on the outside, but the arpuscular type. Remember, the arpuscular is named that because this is an arpuscle, something like that. I don't know how well I spelled that. Okay, folks. All right, that's it for the day. You all have a good weekend. If you've got any questions, you're more than welcome to stick around. If not, I'll stop the recording.